throughout the entire region. The people are interwoven into the fabric of the Adirondack Park. One of the things I love about working in the Adirondacks is, this is really an unrivaled conservation experiment. It's a huge, huge state park. And yet it's not a build a fence and lock people out state park. It's a working landscape. It's really fulfilling to be able to go out in a spectacular natural environment like the lake behind me here and try to understand what's going on in that lake, how its fisheries are changing, what we can do to manage those fisheries to kind of buffer them against many of the environmental changes that are underway, but always cognizant of how people are using the system. There are very few regions of North America where you find brook trout that specialize in living in lakes and ponds. This is the state fish of New York. It's really the emblematic fish for the Adirondacks. So as we think about what the future holds for Adirondack Lakes, one of the primary ways that we maintain the vigor of those fisheries is through stocking fish. That's really the backbone for Adirondack Lake fisheries, is supplementing these populations from hatcheries. As the lakes become ever more inhospitable, which we think is happening all through the Adirondack Park, that raises a challenge for these stocking programs. If the lake is less hospitable, can we pick strains of trout which are better adapted to the changing conditions?
Strains of fish is kind of like a breed of a dog. We wanted to be able to know if we can use environmental DNA to detect these intraspecific differences, so the differences within one species of what fish are here. And that can give us a lot of information about things like where wild populations are, the strains that we have stocked across the Adirondack Park, which ones have persisted or which ones are no longer present. We're wondering if the 6 different strains that we stock in our research program have environmental preferences, so temperature or dissolved oxygen preferences that will allow them to persist or not in a certain lake or in a specific environment. The trouble with climate adaptive stocking is, turns out it's really hard to recapture these fish that we've stocked. And so with environmental DNA, we're able to just collect water, and then I can tell you what strain is there based on the DNA that's there. So using eDNA as a way to know what animals are living here and how they're adapting to the changing environment without having to disturb them or bring them back to the lab. It's such a great tool because it gives us insight into how they are facing all of these unprecedented changes.
Only now the fish are just getting large enough where we can start to assess survival rates and growth rates of those stocked fish. So I'm excited to see how our lab measurements actually correlate with field performance metrics. Here with the Adirondack Fisheries Research Program, we're doing temperature and oxygen trials to assess different populations of brook trout for their resilience to climate change. And this is important because we're finding projections with climate change aren't great for these cold-loving fish species. So understanding how these populations can tolerate these conditions will help us have a more informed and adaptive fisheries management style in the future.
SCALE is the Survey of Climate and Adirondack Lake Ecosystems. Together we've defined an agenda trying to understand the 21st century state of Adirondack Lakes. The opportunity that we're seizing is a follow-up on a 1980s survey. So we can now fast forward 40 years and be able to compare, for instance, what's the species list of fish or of invertebrates in a lake. We can ask questions about how shifts in the climate and other conditions in the Adirondacks have affected the lakes relative to a 1980s benchmark. There's so many things we don't know, and that historical baseline from the 80s provides us just an unparalleled reference point at this kind of scale.
The other thing that's happening is our lakes are browning, and that's because as the Adirondacks recover from acid rain, there are chemical changes happening in the soil. And that means that water that's flowing through the soil is pulling more organic matter into the lakes. Just like tea, how it's stained by tea leaves, our lakes are being more stained by all of the beautiful vegetation you see around us. You wouldn't think much of it, but what happens, especially when combined with climate change, is that it's preventing light penetration from going further down. And that means the deeper parts are remaining colder. And you would think that's a really great thing for all our cold water species that are suffering from warming climate. But the problem is that it causes prolonged stratification. So these two layers aren't mixing. And that means not only is the lake hot on top where cold water species can't live, but there's no oxygen on bottom, so they also can't live here. And that's causing this habitat compression where they have just a small area, or sometimes none at all, where they can live with cold enough temperatures and enough oxygen.
Over the last 10 or 15 years, we've recognized the Adirondacks as sort of the epicenter of this browning phenomenon. So that's a huge problem for the lakes, but it's one that's hidden. So one of the things that I enjoy so much about working both in the Adirondack landscape and with any CASC is we have a shared mission. We all care about trying to find a sustainable future for that place. And that future includes humans, but it also goes beyond humans. In any CASC, it's the same spirit. So we bring together foresters and landscape biologists and wildlife biologists and fisheries scientists and coastal marine researchers. And we all share a passion for the region as a whole and thinking about the way that all the pieces fit together, that we can't fix a problem in one place and ignore the problems in all the other components of that system. It's just a joy to be in a community where you're not the only one that cares. You're working with other people that care, and they bring their talents to the table, and you look for the complementarity and take pleasure in working with other people who have the same big picture motivation, but go about it in a whole different way and have a different toolkit and a different voice. And together, I have great faith that we can solve these problems. But it takes time, it takes effort, and most of all, it takes a team.